Types of probability sampling.
There are the types of probability sampling.
Simple random sampling. In this sampling technique, each population element has an equal chance of being selected into the sample.
The samples are drawn using random number tables or generators. This technique is best used if an accurate, complete and easily accessible sampling frame is available.
By using random numbers, the selection of sample is done without bias, thus making the sample representative of the whole population.
The major disadvantage of this sampling form is that it requires a listing of the population elements. This will take more time to implement.
If the population covers a large random geographical area of selection, then a selected case is likely to be dispersed throughout the area, and will be costly due to high travel expenditure.
Systematic sampling.
In systematic sampling, an element of the population is selected at the beginning, with a random start of a range of 1 to k, and following the sampling fraction is selected for every kth element.
The sampling involves the selection of the sample at regular intervals from the sampling frame.
This sampling technique is simple to design and is flexible. It is easier to use and more efficient than simple random sampling.
It has an added advantage of being easy in determining the sampling distribution of mean or proportion. As it is not necessary to construct a sampling frame, it is less expensive than simple random sampling.
Periodicity within the population may skew the sample and results. For instance, assume the sampling fraction is k equals 4, and the list contains the names of every male followed by a female.
If the first selection is a male, then the sample will contain only male respondents. Consequently, the sample will be biased.
If the population list has a monotonic trend, listing from the smallest to the largest element, a biased estimate will result, based on the starting point.
Stratified sampling.
This is a modification of the random sampling in which the population is divided into two or more mutually exclusive subpopulations or strata, based on one or a number of attributes.
Then, the random selection, simple or systematic, is used on each strata. Results may be weighted and combined.
Stratification can be done based on primary variables under study. The selection of the sample is done either by proportionate stratified sampling, in which the sample is drawn proportionate to the stratum's share of the total population, or by a disproportionate stratified sampling, which is any stratification that departs from the proportionate relationship.
A major advantage of the stratified sampling is that the researcher has control over the sample size in strata.
This control results in increased statistical efficiency because each stratum is homogeneous internally and heterogeneous with other strata.
Moreover, the size of the sample in each stratum provides adequate data to represent and thus analysis of subgroups.
Cluster sampling.
The population is divided into internally heterogeneous subgroups, each with a few elements in it.
The subgroups are selected according to some criterion of ease or availability in data collection. Within subgroups, there is heterogeneity, but between subgroups, there is homogeneity.
Samples are taken from some randomly selected subgroups for further study.
It often involves large samples since there must be sufficient data to stratify or cluster the population.
However, if the method is indiscriminately used, It will increase costs.
Multistage sampling.
This is a sampling method that employs more than one sampling strategy. It usually starts with cluster sampling since it is a method of selecting a group rather than individual elements.
Having selected the groups, clusters, the individual elements representing the groups are determined using other probability techniques mentioned above.
Self-check. What are the factors influencing the choice of the following sampling designs? A probability sample and a non-probability sample. A simple random sample, a cluster sample and a stratified sample.
Non-probability sampling design.
In addressing certain problems, it is not possible to assume that the sample is selected using probability methods, so the sample has to be chosen by some other way.
Non-probability sampling provides a range of alternative techniques based on the researcher's subjective judgments.
Often, field workers carry out the sample selection, so there is greater opportunity that unfairness would enter the sample selection procedure and distort the findings of the study.
Since the probability of selection is not known, the range within which to expected population parameter cannot be estimated.
Often the choice of non-probability sampling is based on practical reasons even though there are technical disadvantages compared to the probability methods.
The use of non-probability sampling can satisfactorily meet the sampling objectives. Sometimes, the true cross-section of the population may not be the objective of the research.
For instance, if there is no desire or need to generalize the results to the population parameters, the sample does not have to be representative of the population.
Another reason for choosing non-probability sampling is the lower cost and time factor. Probability sampling is time-consuming and expensive. If the non-probability sampling is carefully controlled, it can produce acceptable results.
Although probability sampling produces superior results, it is often subject to constant breakdowns in its application. Carelessness in application by the people involved often leads to biased results.
The non-probability technique may be the only feasible method if the total population is not available for the study or not known. In such cases, the sampling frame will not be available to choose the elements.
It may not be possible to determine completely that the respondent of the male questionnaire is actually the person selected or the true cross-section of the population.
Types of non-probability sampling.
Below are the types of non-probability sampling.
Convenience or haphazard sampling.
This is a non-restricted non-probability sampling, in which field workers have the freedom to choose whomever they find. The sample is chosen haphazardly until the required sample size is met.
It is normally the cheapest and easiest to conduct. This method is considered the most useful procedure to test ideas and exploratory research.
The sampling design is considered the least reliable design because there is no control to ensure precision.
Purposive or judgmental sampling.
The sampling form enables selection of sample members that conform to certain criteria. The researcher can use his own judgment to select cases to enable him to answer research questions and meet the objectives.
This form of sample is usually used when the population is small, such as in case study research, and when the main purpose is to select cases that are particularly informative.
It is very useful in the early stages of an exploratory study or in selecting a bias group for screening purposes. The main disadvantage of this design is that the sample may not have the specific criteria, which differ from the criteria of the population.
Quota sampling.
The design is based on the premise sample that will represent the population, as the variability in the sample for various quota variables is the same as that in the population.
The logic is that certain relevant characteristics describe the dimension of the population, thus making the design a type of stratified sample in which the selection of cases within the strata is entirely non-random.
If the sample has the same distribution of these characteristics, then it is quite likely to be a representative of the population.
The quota sampling has several advantages over the probability sampling. It is in particular less costly and can be set up rather quickly.
It does not require a sampling frame and may be the only technique that can be used if other techniques are not available.
It is most useful if the population is large, since the sample size is governed by the need to have sufficient responses in each quota to enable subsequent analyses to be undertaken. Hence the total sample size may be more than 2,000.
A major weakness of the quota sampling design is that the assumption of the quota being representative is arguable, as there is no assurance that each variable under study represents the population characteristics.
Available data used as a basis for the determination of quota may be outdated or inaccurate, thus without relevant or sensible quotas, data collected may be biased.
The number of control variables that are used may be limited and is often left to the choice of the field workers.
Snowball sampling.
This design is usually used when the respondents are difficult to identify and located through some referrals from people who know them.
The respondent may or may not be chosen initially through probability. The initial individuals are used to locate other individuals who have similar characteristics, and who, in turn, identify others.
The referral approach can help to reach particularly hard-to-find respondents. However, it may get only individuals similar in characteristics to the introducers.
The design may result in a highly homogeneous group.
Activity. In a situation where the respondents live in rural areas, what is the most effective type of sampling that can be used?
Sample size.
The sample size is more often than not determined by judgment as well as calculation. In many cases, the types of statistical analysis used would determine the minimum sample size for each individual category.
As a rule of thumb, a sample size of 30 is the smallest number in each category within the overall sample that is acceptable.
The sample size is the number of elements to be studied in the research project. Determining the size is one of the great challenges of many junior researchers.
Some of the major considerations in sample size determination are namely:
importance of the decision, larger and representative sample size for important decisions.
The nature of the research, smaller size for exploratory.
The number of variables, larger size if more variables are involved.
The nature of data analysis, detailed and sophisticated statistical analysis require large random samples, and
resource availability. The size can be determined statistically or non-statistically.
Qualitative approach.
Determining the sample size involves both qualitative and quantitative consideration. There are a few qualitative factors in determining sample size as discussed in the earlier part of this topic.
One of the commonly referred rules of thumb for determining sample sizes, especially in the exploratory research, is the user-friendly model of Krejci and Morgan.
Many also have used their suggestion in determining sizes in certain phases of the probability sampling, for example, determining the size of each stratum in a stratified sampling.
They simplified the sample size determination based on the respective target population sizes.
Table. Some of the suggested sizes.
Sample sizes by target population sizes.